Hello everybody, I am Dr. Sandeep Choudhary from Department of Advertising and Marketing Management from Patna Women's College, Autonomous. Today we are going to discuss about sampling in research. So first of all, let's understand what is it? What is sampling? Sampling is a process in statistical analysis where researchers take a predetermined number of observations from a larger population. Sampling means selecting the group that you will actually collect data from in your research. For example, if you are researching the opinions of students in your university, you could survey a sample of 100 students. In statistics, sampling allows you to test a hypothesis about the characteristics of a population. Now the question arises, why sampling? So in many studies, it is not possible for the researchers to test the entire elements of the population. So, for such studies, sampling is the only method by which studies can be done, and based on the outcome of the samples, the generalizations about the nature or trait of the entire population is made. For example, in medical science, to make diagnosis of diseases through tests like blood test, only sampling could be used, and not the entire blood from the body could be extracted for the test. Even in cooking food also, we take to test only few samples to decide whether the food is cooked or not. Similarly, in researches for the large populations, it is justifiable to use sampling only. Now, the question arises, is there any alternative? So yes, there is an alternative. The alternative of sampling is only census method of study, in which entire elements of the population is included in studies. There are several studies for which only census can be used. For example, for counting the number of people living in a place, only census can be used because number of people living in a place can't be guessed or counted on the basis of sampling. Rather, each element should be included. But again, for many studies, researchers don't have sufficient time and money to do census. Now let's talk about sampling technique. There are basically two types of sampling technique. First, probability sampling and non-probability sampling. Probability sampling is also known as random sampling or chance sampling. Under this sampling, every item of the universe has an equal chance of inclusion in the sample. Here, it is blind chance alone that determines whether one item or the other is selected. This applies to sampling without replacement. That is, once an item is selected for the sample, it cannot appear in the sample again. Now, non-probability sampling. Non-probability sampling is also known by different names like deliberate sampling, purposive sampling, and judgment sampling. In this type of sampling, items for the sample are selected deliberately by the researcher. His choice concerning the items remains supreme. But in such kind of sampling, personal element has a great chance of entering into the selection of the sample. It means the investigator may select a sample which will yield results favorable to his points of view, and if that happens, the entire inquiry or study may get spoiled. Now let's talk about types of probability sampling. The first one is simple random sampling. Here, every element has an equal chance of getting selected to be the part sample. Second one is systematic sampling. Only the first unit is selected randomly and remaining on fixed intervals. The third type is stratified sampling. Here the population is subdivided into strata, then we select items from each stratum to constitute a sample. The next one is cluster sampling. Here the population is divided into small subdivisions called clusters. Then some of these clusters are randomly selected. The next one is area sampling. If clusters happen to be in some geographic subdivisions, then it is area sampling. Now let's talk about types of non-probability sampling. Here the first one is convenience sampling. As the name suggests, here the samples are selected on the basis of availability. This is good when availability of sample is rare and costly. Next one is purposive sampling. Here the selection of samples is done whoever seems to be most appropriate for study. The next one is snowball sampling. This occurs by generating leads from the samples. This is used where the population is completely unknown. The next one is quota sampling. Here the first strata are formed, then sample units are selected until you reach your quota. That's it. Thank you everybody.